Hello, anatomy friends. This is Dr. Alsup, and in this video, we are going to be discussing the muscle cells associated with skeletal muscle. And you see here, I have three different names that I'll be using interchangeably that all mean the same thing. So we are talking again about the skeletal muscle cell, but sometimes you hear this referred to as a myofiber. And then sometimes you hear this referred to as a muscle fiber, and that has to do with the relatively long size of a skeletal muscle cell. So we have much to do here, so let's get going. As you can see, I have 6 learning objectives that we are going to work through. This is a little bit more complex of a topic, so we're going to jump right in. And we're going to start with how do we even get these comparatively long muscle cells? Well, during embryonic development, each skeletal muscle fiber is formed by the fusion of around 100 or more myoblasts, which are just small mesodermal cells, and they're going to fuse up to 100 of them to form what we refer to as those muscle cells or those myofibers. And because they are 100 plus different cells that are fusing together, in that myofiber, you're going to have 100 plus nuclei because each one of those myoblasts had their own nuclei. So the muscle cell is going to be multinucleate cell. Once these myoblasts have fused, the muscle fiber is considered mature and can no longer undergo cell division. And that's unique. So in other words, the number of skeletal muscle cells you have are set before you're even born. But that doesn't mean that skeletal muscle can't or won't change. We know that that's not true. You can actually have dramatic muscle growth after birth and maybe a little less so throughout life. And this is going to be via muscular hypertrophy rather than hyperplasia. So muscular hypertrophy would be the enlargement of those existing muscle fibers, whereas muscular hyperplasia would be an increase in the number of muscle fibers. So again, it is muscular hypertrophy that occurs after birth. And what happens here is you're going to have an increased production of myofibrils, which we will discuss in detail shortly, but it's going to be the major component within muscle cells. And you can have other organelles that can increase as well. And this is caused by forceful, repetitive muscular activities. Think of strength training. We know that that will make muscles get larger, and that's how it's doing it. And if you have more myofibrils, you're capable of more forceful contractions. During childhood, hormones, particularly human growth hormones, will stimulate an increase in the size of those skeletal muscle fibers. But conversely, you can have a progressive loss of myofibrils when muscles are not used or the nerve supply to the muscle is disrupted, and this will lead to a decrease in size and strength of the muscle, and that's going to be referred to as atrophy or muscular atrophy. All right, there are numerous components to this relatively long muscle cell that again is often called a muscle fiber. If you see sarc, S-A-R-C, that's going to mean flesh or muscle. So often we just plop a sarc on a more general word for the organelles of a cell. So the sarcolemma is the plasma membrane of a muscle fiber. So that cell membrane on the outermost boundary of the cell. And this is a really important area for excitation and conductivity of a muscle cell. This image on the right shows that there are nuclei that are pressed right up against the inside of the sarcolemma. And remember, there are multiple nuclei associated with each cell. The sarcolemma will also have infoldings that will kind of tunnel into the inside or the deeper portions of the cell. And these are called transverse or often shortened to T tubules. And these tubules are important in getting an action potential into the deeper parts of the cell, which can lead to calcium release. And there'll be more to come on that calcium release in a second. The sarcoplasm is just the cytoplasm of a skeletal muscle cell, so it's enclosed by that sarcolemma. And like other cells, the sarcoplasm is filled with numerous types of organelles. By far, its major component is going to be what's referred to as myofibrils. In fact, in this illustration on the right, it looks like myofibrils almost completely fill up the sarcoplasm. And that's kind of true. They are truly all over the place. But there are other organelles. We mentioned the nuclei. You can also have mitochondria and other structures as well. So it's a very dense, filled-in region. One of those other organelles is going to be the sarcoplasmic reticulum, often just abbreviated to SR because that's a rather long term. This will form a complicated network around the myofibrils. So it's going to be this complicated network in this illustration here. And the SR is just the smooth endoplasmic reticulum of a skeletal muscle cell. And through the SR, you're going to have these dilated sacs called terminal cisternae. And these are going to be very, very closely associated to a T-tubule that we just discussed of the sarcolemma. In fact, one T-tubule plus two terminal cisternae is going to form a triad, which is an important component of the muscle excitation contraction coupling step of muscle contraction. And the sarcoplasmic reticulum is a reservoir of calcium ions, specifically in a relaxed muscle. And when a nerve impulse initiates an action potential in the sarcolemma, this will travel down the T-tubule and then signal the SR to open its gated channels and release a flood of calcium into the sarcoplasm, which, as we know, is of course filled with all of those myofibrils, which are the keys to contraction. So muscle contraction cannot occur without this calcium, and that calcium is hanging out within a sarcoplasmic reticulum in a relaxed muscle. So let's get into some actual details about those threads within the sarcoplasm, aka the myofibrils. And these are just long protein cords that fill up most of the cell, and each myofibril will actually extend the entire length of a cell. And it is within these myofibrils that those keys to contraction lie, the myofilaments and the proteins that make those. Each myofibril is going to be composed of smaller myofilaments. So let's talk about some of the more important myofilaments. The two major myofilaments are thick and thin filaments, and these are composed primarily of the contractile proteins actin and myosin, depending on the filament type. And contractile proteins are these are the proteins that are converting ATP's chemical energy into the actual mechanical energy of motion. Thick filament is going to be composed of hundreds of molecules of the protein myosin. Myosin molecules are shaped like two golf clubs that are interwoven with two kind of globular heads that are just going to be projecting from the tail. So a thick filament is going to be a bundle of all these golf clubs or myosin proteins. Thin filament is a little more complicated. It's composed of primarily actin proteins. So if a myosin is like two golf clubs that are wound up together, the weird comparison that we're going to use for actin is it's like two chains of pearls or a beaded necklace that's kind of interwoven. And each one of these actin molecules will have an active site to bind the head of myosin during the contraction process. Thin filaments also have what's called regulatory proteins too. So regulatory proteins help switch off and on the contraction process. And tropomyosin is the major protein that is associated with thin filament. So when a muscle is relaxed, each tropomyosin is going to block the active sites of around 6+ actins. And you can see this in the illustration on the bottom right. And this prevents that myosin from binding to actin, which is what you want during a relaxed state. But each one of these tropomyosin molecules will have a smaller protein bound to it called troponin, which has a receptor for calcium. And so when a calcium attaches to the troponin, say after the release from the sarcoplasmic reticulum, it causes the shape of the troponin-tropomyosin complex to change. And this will cause tropomyosin to move away from the actin, and that will expose their active binding sites for those myosin heads. Now, I do want to note that myosin and actin are not unique to just muscle cells. They do occur in other cells with different functions, such as transport of intracellular materials or cellular mobility. And even within a muscle cell, different forms of actin will have different functions as well. But we're focusing in on the portions that are important with the contraction process. So thin and thick filaments are definitely the most discussed, but there are other myofilaments, other proteins we want to make sure we understand. There are elastic filaments that are made up primarily of the huge springy protein called titin or titin. This will run through the core of a thick filament and anchor to structures called a Z disc at one end and the M line on the other. And so it serves to stabilize and center the thick filaments between thin filaments mainly, but it also plays an important role in preventing overstretching. There are about 7 other accessory proteins associated with thick and thin filaments. One of the most clinically relevant ones is going to be dystrophin, which is a large protein located between the outermost myofilaments and the sarcolemma. And what dystrophin will do is it will link actin filaments to peripheral proteins on the inner surface of the sarcolemma, which through a series of links actually leads all the way to the fibrous endometrium, which is the connective tissue that surrounds individual muscle cells, if you recall. So when those thin filaments move, they're pulling on the dystrophin, and this ultimately pulls on the extracellular connective tissue, leading eventually to a tendon, because remember, all of the connective tissue is continuous. So you can think of dystrophin as serving as a shock absorber to protect the sarcolemma during the mechanical stresses of contraction. Now, dystrophin may sound familiar to you if you have ever heard of muscular dystrophy, which includes a range of inherited genetic disorders, which prevents the body from producing dystrophin, or it produces a defective form of dystrophin. And this can lead to progressive and irreversible muscle weakness and atrophy because the cell membrane becomes so fragile and prone to tears without that protection of dystrophin, and that could lead to cell death. Okay, let's talk about striations. We mentioned earlier on that skeletal muscle appears striated microscopically. So what's actually causing these striations? One initial thing people will notice when looking at striations is that some areas will appear darker and some areas will appear lighter. And we're going to focus on this top part of the illustration here. This is representing a relaxed muscle, whereas this is a contracted muscle. We'll talk a little bit more about this in an upcoming slide, but we're going to focus on what it looks like in a relaxed muscle. And we have what's referred to as the dark A band. I remember this because there is an A in dark. So the A band is going to be dark. And this A band has thick filaments lying side by side, But also part of the A band is where the thick and the thin filaments are going to overlap. And that portion of the A band is going to appear even darker. The middle of the A band has a lighter region, although it's still considered part of the dark portion, called the H zone, where the thin filaments do not reach in the relaxed muscle. All right, the I band is going to be light, and there is an I in light, so that helps in terms of remembering that. This is the area that the thick filament does not extend in a relaxed muscle. And each I band is importantly bisected by a narrow and dark Z disc, which provides anchorage for the thin as well as those elastic filaments we talked about in the previous slide. Each segment from one Z disc to another is called a sarcomere. And this is the sarcomere is the contractile unit of a muscle fiber. So a muscle will shorten because its individual sarcomeres will shorten and pull the Z discs closer to one another, which you can see on this image on the bottom. So the Z discs are going to become closer together during the contraction process. And this brings us to the model that describes the contraction of a muscle, and this is called the sliding filament mechanism. And it's important to note that a myofilament, those thick and those thin filaments, they don't actually shorten. Rather, the thin filaments slide inward and meet at the center of a sarcomere, which is shown in this illustration on the bottom left. So it's the amount of overlap between the filaments that change. And shortening of the individual sarcomeres will cause shortening of the whole muscle fiber, which in turn leads to shortening of the entire muscle. All right, excellent. That was a long one with a lot of info. So take your time in terms of reviewing those learning objectives. But let's work through a practice question together. Which structures of a myofiber stores calcium in a relaxed muscle? What do you think? Well, actin and dystrophin are not, these are proteins. They're very important in terms of the muscle, but this isn't what's storing calcium. So let's mark these off. These are not correct. Sarcolemma is the plasma membrane, so the outside portion of the cell. That's not storing calcium either. So let's mark this one off too. And that leaves sarcoplasm and sarcoplasmic reticulum, which both can have calcium. But the key in the stem here is in a relaxed muscle. So the relaxed is key. So the sarcoplasmic reticulum is storing calcium in a relaxed muscle. All right, so that is correct here. It can release calcium during the contraction process into the sarcoplasm, but again, that is when the contraction process is occurring. So if it is a relaxed muscle, the calcium is being stored in the sarcoplasmic reticulum, so E is the correct answer. Excellent. Thank you so much for your time and attention.